So good morning, students. Am I audible?
Yes, ma'am.
Okay. Okay, so we'll continue with the previous class that was on properties of protein and structure of proteins. So Mohit Yadav is there. Mohit Yadav, roll number 629. Then Hina Yadav.
Present, ma'am.
Okay, okay. What about Mohit Yadav?
I don't know.
So Hina Yadav, did you attend the class last time?
Yes, ma'am.
Okay, so can you tell me what are the different levels of structural organization of protein?
Primary structure, secondary structure, tertiary structure, and quaternary.
Quaternary structure, no? So how will you define primary structure?
On the basis of sequencing and number of amino acids
Okay, how these
that
amino
link
acids are linked in a polypeptide chain? What is the bond?
peptide bond,
Okay, good. Then what are the different types of secondary structure that you have learned?
alpha helix and beta plated chain,
Okay, good.
loops.
And yeah, loops and turns, no? Okay. So any,
Yeah.
what do you mean by tertiary structure?
It is a 3D arrangement, static relationship in 3D arrangement.
Okay. And can you name any protein having a quaternary structure?
Hemoglobin.
Okay, good. ठीक है. Okay, so we'll continue with the previous class and we'll, in relation to the structure of proteins, when the structure of protein is altered, we get a disease known as Prions disease. Have you heard, any one of you, about Prions disease? Okay, so Prions disease, it occurs because of the misfolded protein. There is a defect in the structural organization of protein. We get misfolded protein in Prions disease. It is also known as spongiform encephalitis. Okay, because there are spongiform changes in the brain cells. That's why it is known as spongiform encephalitis. Clear? So it is a neurodegenerative. It comes as a short note, Prions disease. So remember, Prions disease is a neurodegenerative disease which is caused by misfolded proteins. Clear? So since it affects the brain, so all the symptoms are related to the brain, like there may be dementia. What do you mean by dementia? Dementia means loss of memory, isn't it? Then there will be a loss of coordination, then encephalitis. So this Prions disease, remember, it is caused because of the infectious protein particle. So these protein particles are passed in different species. For example, when these protein particles are present in sheep, the disease is known as Carpes disease. Okay, when the protein particles moves from the sheep to the cow, then it gives to a condition known as mad cow disease, also known as bovine spongiform encephalitis. So the same protein particles, when it moves from the cow to the human, it results in a disease known as Creutzfeldt-Jacobs disease. Okay, so these are a few examples of Prions disease. So the disease name is different in different species. So what is the causative agent here? It is a prion protein, okay, PRP protein, which is encoded by a PRP gene. Okay, this gene PRP gene, it encodes the protein PRP protein. So remember this PRP protein, it exists in two conformations. One is PRPC, which is found in the normal cells, and PRPSC, which is the infectious prion particle, okay, which causes the Prions disease. So this PRPSC, it is resistant to protease enzyme, okay, and PRPC is converted to PRPC after contact with PRPC. So when this prion protein, this infectious PRPSC protein comes from the sheep and it enters the human being, it converts the normal PRPC protein into a abnormal PRPSC protein, which is protease resistant, and it results in misfolded protein, resulting in a disease known as the Creutzfeldt-Jacobs disease in humans. Any confusion? Prions disease. So this Prions disease, remember, it is because of the infectious protein particle, that is the PRP protein. Okay, so with this PRP protein, it exists in two conformation. One is PRPC, which is a normal protein, and when PRPSC is converted into PRPSC, okay, then it becomes an abnormal protein. Okay, so this PRPSC, SC stands for SCARP. So PRPSC, it is the protein which causes the protein to misfold. Clear? And the name of the disease is different in different species. In cow, remember, it is known as mad cow's disease. In sheep, it is known as Carpy's disease. And in human, it is known as Creutzfeldt-Jacob's disease. Clear? Now let us come to protein sequencing. We will come to the primary structure of the protein. How do we know the sequence of amino acids that are present in the polypeptide chain? So in order to know the sequence of the protein in a polypeptide chain, first of all, we have to isolate the protein. For example, if you want to know the sequence of insulin, which is present in the plasma, first you have to isolate the insulin from the blood, isn't it? But in the blood, there are different kinds of protein that are present, like albumin, globulin, fibrinogen, then various clothing factors, hormones, enzymes, etc. So you want to isolate the insulin from these different types of proteins which are present in a blood. So what are the different procedures by which you can isolate this insulin? It may be by ultracentrifugation, differential ultracentrifugation, because all these proteins have got the different molecular weight. So they will sediment according to their molecular weight. Okay. So they may be separated by differential ultracentrifugation. Once they are separated, then they have to be purified. And the purification of the protein, remember, it is based on the solubility and the molecular size. So these techniques I will not go into detail because it is a very long chapter. So remember, there are different techniques by which you can isolate a purified protein, like adsorption chromatography, partition chromatography, then gel exclusion chromatography, okay, not isoelectric focusing, and HPLC. So this for adsorption, partition, gel exclusion, and HPLC. What is HPLC stands for? Anyone? Any student who knows what is the full form of HPLC? Vivek?
Ma'am, no idea, ma'am.
No idea. Anup? Anup, you know what is the full form of HPLC?
No, ma'am, I forgot.
Okay. So HPLC stands for high performance liquid chromatography. Clear? High performance, it is also known as high pressure or high performance liquid chromatography. Clear? So if you want to purify a protein, the protein can be purified by different techniques like adsorption chromatography, then partition chromatography, gel exclusion chromatography, and high performance liquid chromatography. In practical, have you been taught about paper chromatography in your practical classes?
No, ma'am.
No. Okay. Okay. So you will be taught in practical class how to perform paper chromatography. Okay. So what happens in paper chromatography is actually we take a sheet of filter paper. Okay. This is a Wattman filter paper. We take a sheet of filter paper, then we make a point in the filter paper at one end, and here in this point we add the different amino acid mixtures. Okay. Now what we'll do next is we will soak this filter paper in a tank which consists of a solvent. Okay, the solvent contains a polar as well as non-polar mixture. So these amino acids will migrate along with the polar solvent, and they will migrate at different distance depending upon their polarity. Okay, those amino acids which are non-polar will move far away from this spot. On the other hand, the amino acids which are polar will stay nearby this spot. So they will migrate at a different distance, and by calculating this distance, you will be able to know which amino acid is it. Okay, each and every amino acid has got a standard RF value, relative front value. So by knowing the relative front value of these different amino acids, we will be able to identify what type of amino acid is it, whether it is a glycine, alanine, or phenylalanine, whatever it may be. So each of these amino acid will have their own standard RF value. So after performing paper chromatography, we will measure the distance of these amino acids from the point of application, and by measuring the distance of this amino acid, the distance traveled by the amino acid, we will be able to calculate the RF value. Okay, and so each and every amino acid will have its own standard RF value. So this is some just gist of paper chromatography. The details will be taught in your practical classes. Okay, this one is very important and it is difficult to understand, so please try to listen carefully. So if you want to know the sequence of amino acid, for example, this is a structure of insulin, isn't it? Insulin has got two chains, the A chain and the B chain. The A chain, it has got 21 amino acid. The B chain has got 30 amino acid. Then there is a disulfide linkage, interchain disulfide linkage between the cysteine residues which is present in the position number 7 in both alpha and beta chain, and the cysteine residue between 19 and 20. Okay, as well as there is an intra-chain disulfide bond between 6 and 9. So if you want to know what is the sequence of amino acid in the insulin molecule, first you have to separate this A chain and B chain, isn't it? First you have to separate this A chain and B chain, and then you will be sequencing one by one. The first here in this A chain is glycine, isn't it? After glycine comes isoleucine, then valine, then glutamate, glutamine. So this sequence, how will we know in practically that what is the sequence of any protein? Okay, so first if you want to know the sequence of a protein, what you have to do, suppose this is a hemoglobin molecule having a quaternary structure, having 4 polypeptide chain, 2 alpha and 2 beta chains. Now all these polypeptide chains will be connected by non-covalent bonds, isn't it? So first you have to break these non-covalent bonds in order to separate these different monomeric units. So this non-covalent bonds is hydrolyzed by adding either urea or guanidine hydrochloride. Okay. And if you want to break the disulfide bond, for example, in the insulin molecule, if you want to break the disulfide bond, you have to add another molecule that is beta-mercaptoethanol. Okay, so by treating the protein molecule with these different reagents like urea, guanidine hydrochloride, or mercaptoethanol, we will separate the quaternary structure and convert it into a primary structure. Clear? Any confusion? Have Vivek bolo?
No, ma'am.
First, what is the first step to sequence the protein?
To break the non-covalent bond.
Non-covalent bond present where?
In between the quaternary structure.
Yeah, in between the quaternary structure. Suppose a protein molecule, no, any protein molecule, it may be having a quaternary structure or it may be having a structure only up to the tertiary level. But by chance, if it has got a quaternary structure, then first you have to break the non-covalent bonds in order to separate the monomeric unit, isn't it? So this is done by adding reagents. You have to remember these reagents. You can go through the textbooks again. Okay, by adding urea and guanidine hydrochloride, this will break the non-covalent bond. And if any disulfide bond is present in between the subunits, then it will be hydrolyzed by adding beta-mercaptoethanol. So once these quaternary structures are hydrolyzed, then you get a primary structure. Then the next step is to know the amino acid composition. We will not go towards the amino acid sequencing first. We just want to see what are the amino acids that are present in a hemoglobin molecule. Okay, so we have already broken the quaternary structure into a primary structure. Now we will see what are the amino acids which are present in the primary structure, but we are not interested in sequence now. So what we will do is first you have to identify how many polypeptide chain is present in a protein. For example, if hemoglobin, we are having 4 polypeptide chain, isn't it? So how do we know that this protein is having a 3 polypeptide chain or 2 or 4? This is done by adding a reagent or a chemical known as the densyl chloride. So what this densyl chloride will do, it will go and react with the amino acid which is present in the N-terminal end of each polypeptide chain. Clear? Densyl chloride will react with the amino acid that is present in the N-terminal end of each polypeptide chain to form the densyl amino acid. So by chance, if you treat the hemoglobin molecule with the densyl chloride, how many densyl amino acids will be formed? Anu, bolo?
Ma'am, the number of polypeptide chains will form the number of densyl amino acids.
So how many densyl amino acids will be formed if you treat hemoglobin with densyl chloride?
4, ma'am.
Yes, 4. Okay, CK, creatine kinase, how many will be formed?
3, ma'am.
CK is having only 2 polypeptide chain, no? MB, no? No?
Okay.
Yeah, only 2. So depending on the number of the polypeptide chains, the number of amino acids will differ in different proteins. Clear?
Yes, ma'am.
Okay, now you have identified how many number of polypeptide chains are present in the protein molecule. Now in the next step, the third step, you want to just know what are the amino acids present in this polypeptide chain. So what you will do, you will hydrolyze all these polypeptide chains with 6 normal hydrochloric acid. Okay, then heat it at 110 degrees Celsius for about 18 to 36 hours in anaerobic condition, and all the peptide bonds will be hydrolyzed to form different amino acids. Okay, now you will subject these amino acids to paper chromatography, and depending upon the RF value, okay, by calculating the RF value for each and every spot that is present on the paper, you will be able to identify what is the type of amino acid that is present and how many numbers of amino acids are present. So the first step is to treat the protein with urea or guanidine hydrochloride to break the non-covalent bonds, and beta mercaptoethanol to break the disulfide bonds. Then you get the individual polypeptide chains. Then treat those polypeptide chain with densyl chloride to form the densyl amino acid to identify how many numbers of polypeptide chain is present in a protein. Okay, in the third step, you hydrolyze all those polypeptide chains with 6 normal hydrochloric acid. Then you get amino acid, isn't it? The hydrolyzed product will be amino acids. Now you have to identify these amino acids by performing a paper chromatography, and by calculating the RF value you will be able to know how many numbers of amino acid are present in that protein and what are the types of amino acid present in that protein. Clear? Any confusion?
No, ma'am.
Okay, okay. So this is regarding how to know the composition of the amino acid that is present in a protein. Next is how to identify what is the sequence of this amino acid in a protein. For example, you want to know the sequence of this amino acid in the A chain of the insulin. So what we'll do is, first of all, you will break those complex protein into a monomeric unit by treating them with urea, guanidine hydrochloride and beta mercaptoethanol. Then you will treat that polypeptide chain with densyl chloride. Okay. In the next step, you will not hydrolyze with 6 normal hydrochloric acid. What you will do is, you will hydrolyze this protein. Okay, remember one thing, insulin was the first protein which was sequenced by Frederick Sanger, for which he received a Nobel Prize in 1958. So in the Viva, if they ask, name the first protein which was crystallized and which was sequenced, remember it is insulin. Clear? So if you want to sequence the protein, first what you have to do is you have to break this polypeptide chain, okay, into different fragments. So if you want to break this polypeptide chains in between the polypeptide chain, you will use an enzyme known as endopeptidase. Okay, endopeptidase will break the peptide bond from inside. Exopeptidases will break the peptide bond from either the N-terminal or C-terminal, isn't it? So if it breaks the peptide bond from the N-terminal, it is known as N-terminal exopeptidase, and if it is from the C-terminal, it is C-terminal exopeptidase. Okay, so you want to break this polypeptide chain in between to form various fragments, so you will require an endopeptidase. Example of endopeptidase is trypsin. Okay, trypsin will break the peptide bond which is formed by the carboxyl group of arginine and lysine. Similarly, chymotrypsin will break the carboxyl group, okay, will break the peptide bond which is formed by the carboxyl group of aromatic amino acid. Remember this one. Okay, sometimes it is very difficult to remember. Trypsin, in which type of amino acids it will act? It will act on the basic amino acid, that is arginine and lysine. Clear? Chymotrypsin will act on aromatic amino acid. Then you note down the third one. Okay, I have not written here. Cyanogen. Okay, cyanogen bromide. Cyanogen bromide will break the peptide bond which is formed by methionine. Okay, cyanogen bromide will form the peptide bond which is formed by methionine. So you can break these peptide bonds which are present inside the polypeptide chain by using either the endopeptidases, or you can break by using chemicals like cyanogen bromide. Clear? Then when you break this polypeptide chain, what you'll get? You will get different fragments of different sizes, and some of these fragments will have an overlapping fragment, isn't it? Suppose this is one fragment and this is the second fragment. You can see that some portion of the first and second fragment, they overlap, and this is known as the overlapping fragment. Clear? So we will treat this polypeptide chain with different endopeptidases or by using chemicals to get the overlapping fragment. And each and every overlapping fragment will have the N-terminal end as well as C-terminal end. Yes or no? Any confusion? Anup?
No, ma'am.
Okay. So each and every fragment will have the, each and every overlapping fragment will have the overlapping amino acids. And each overlapping fragment will have the N-terminal end and the C-terminal end. Now what we will do, we will try to sequence each and every overlapping fragment. Okay. Now you can sequence it either from the N-terminal end or either from the C-terminal end. Suppose this is the N-terminal end, this is the C-terminal end. Okay. We'll go back here again. This is the N-terminal end because the numbering always starts from the N-terminal end, isn't it? So glycine, this is the N-terminal end. You can sequence either from the N-terminal side or from the C-terminal side. So if you want to sequence the protein from the N-terminal side, we can do by 3 methods. Number 1 is by using a Sanger's reagent. You have to remember the name of the Sanger's reagent. This is known as difluoronitrobenzene. This is the Sanger's reagent, okay, difluoronitrobenzene. So when you add this difluoronitrobenzene, it will go and react with the N-terminal amino acid and it will form the dinitrophenyl amino acid derivative. Okay, once it forms the dinitrophenyl amino acid derivative, it can be hydrolyzed and the separated amino acid can be identified by paper chromatography. Clear? Then number 2 is you can use the Edmonds degradation technique, which uses the chemical that is phenyl isothiocyanate. So phenyl isothiocyanate will react with the N-terminal amino acid and it will form the phenyl thiohydantoin derivative. Clear? Or you can use the densyl chloride like in the beginning. Densyl chloride will act with the N-terminal amino acid and it will form the densyl amino acid. So by 3 techniques, you can identify the N-terminal amino acid and you can go on sequencing one by one. Okay, for example, if you add the Edmonds reagent, that is phenyl isothiocyanate, it will go and first go and react with the glycine molecule. Okay, then it will form the phenyl thiohydantoin derivative of glycine, and once it forms this derivative, the glycine, this peptide bond here, this peptide bond, it can be hydrolyzed and this glycine which is separated can be identified by paper chromatography. Okay, then in the next step again you add the Edmonds reagent which will form a complex with isoleucine and then again separate, hydrolyze this peptide bond and then identify the isoleucine. So this step is repeated and then one by one you will be able to sequence the amino acid from the N-terminal end. But if you want to sequence the whole polypeptide chain by using either Sanger's reagent, Edmonds degradation or densyl chloride, it will take so much of time. So what we need to do is we have to break this polypeptide chain into different overlapping fragments by using either endopeptidase or by using chemicals like cyanogen bromide. Clear? Then next is you can sequence the polypeptide chain from the C-terminal end. Okay, so again we'll just repeat from the N-terminal end. This one is the Sanger's reagent, which is fluorodinitrobenzene. Then it forms the dinitrophenyl derivative with the N-terminal amino acid. Then the Edmonds degradation uses the phenyl isothiocyanate, which is converted to phenyl thiohydantoin derivative. Okay, first of all you label this first amino acid with phenyl thioisocyanate. It will form phenyl thiohydantoin derivative, hydrolyze the peptide bond and identify. Then you will label the second amino acid and the step is carried like that. Okay, so densyl, the third way how you can identify the N-terminal amino acid is by adding the reagent densyl chloride. Okay, then we can identify the sequence of the amino acid from the C-terminal end by using carboxypeptidases. Okay. So carboxypeptidases is exopeptidase. It acts from the carboxy terminal end. So you go on hydrolyzing the amino acid one by one from the carboxy terminal end. The released amino acid you identify with paper chromatography. Okay, then in the next step, what you can do, you have identified the sequence of all the overlapping fragment. Then you combine those overlapping sequence of this overlapping fragment, and you can elucidate the complete sequence of the amino acid present in the polypeptide chain. Okay, so this you can sequence the amino acid either from the N terminal or from the C terminal end by using different reagents. But if you use if you do the sequencing without breaking the polypeptide chain, it will take lots of time. So you have to break the polypeptide chain to get the overlapping fragment. Each and every overlapping fragment will have an amino acid sequence, and the carboxy terminal sequence will have a carboxy terminal end and the amino terminal end. And then you sequence each and every overlapping fragment either from the N terminal end or C terminal end by using different reagents. And then after that, what you will do is you will try to elucidate the complete sequence of the protein by seeing the sequence of this overlapping fragment. Clear? So nowadays, sequencing can be done in an automated sequencer, and this automated sequencer uses the Edmonds degradation technique, and the amino acid which comes out are identified by HPLC system, that is high performance liquid chromatography. Clear, students?
Yes, ma'am.
Okay. Chandani is there, no?
Yes, ma'am.
So can you tell me, just repeat whatever you know. You may not be able to, this one, you may not be clear, but whatever you know, can you just repeat whatever I have told? How do you sequence? Okay, you don't want to know the amino acid composition, you want to sequence one protein. So how do you do it?
Ma'am, firstly, we will break the polypeptide chain
Okay.
so that we will get the primary structure.
Okay.
After that, ma'am, we will break the non-covalent bonds by adding urea
No.
or
Yeah, but in order to get the primary structure from the quaternary structure, you have to add the urea first, no? You have to add urea or guanidine hydrochloride or maybe beta mercaptopethanol to convert the quaternary structure into a primary structure. Okay? After that, what we'll do?
after that, we will in step 2 we will add densyl fluoride.
Okay. Why do you add densyl chloride?
And then,
Why do you add densyl chloride?
ma'am, I can't recall, but I wrote that in step 2.
You add densyl chloride in order to identify how many number of polypeptide chain is present in the protein. For example, if it is a hemoglobin molecule, it will have 4 polypeptide chain, isn't it? So if you add densyl chloride, it will form 4 densyl amino acid. So by knowing the number of the densyl amino acid, you will be able to know how many polypeptide chains are present in the protein. Clear? Okay, then in the third step?
Then, ma'am, we will do complete hydrolysis of amino acids.
That is to know the composition. But if you want to know the sequence, what you will do? What you will do in the next?
Paper chromatography.
No, not paper chromatography. What you will do if you want to know the sequence of the amino acid that is present in the polypeptide chain. For example, if you want to know the sequence of amino acid which is present in the A chain of insulin, what you will do?
We can determine the sequence either by starting from N-terminal amino acid or from C-terminal. For C-terminal, we will add carboxypeptidase.
Okay. For N-terminal?
And for N-terminal, there are 3 things that we can add. Firstly,
just name the technique. First is Sanger's technique, no?
Sanger's reagent. Second is Edmonds degradation.
Okay.
And third is densyl fluoride.
Yeah, densyl chloride. Okay. So before sequencing the polypeptide chain from the N terminal or from the C terminal end, what you have to do? You have to break the polypeptide chain into different fragments by using endopeptidases to get the overlapping fragment. Okay. So you have to break the whole polypeptide chain by using endopeptidases or various chemicals to get different fragments. Once you get different overlapping fragments, you can sequence those overlapping fragments either from the N terminal end or from the C terminal end by using different techniques. Clear? Chandani? Please, please read it again in the textbook, then the things will get this one clear. Okay, that's all regarding sequencing of protein. How do you identify, because it is very easy to say, isn't it, the protein is having alpha helix or beta pleated sheet. How do you identify this type of structures, in a computer or by any this one? So you can identify where the secondary and tertiary structures by using just know the name, okay, by using X-ray crystallography, the name of the techniques, please write it down. So you can identify or study the secondary or tertiary structure of a protein by using technologies like X-ray crystallography, diffraction study, UV spectroscopy, or nuclear magnetic resonance study. Okay, and if you want to identify the molecular weight of the protein, this is done by a technique known as mass spectroscopy. Please write it down. The molecular weight of the protein can be determined by using mass spectroscopy. The secondary or tertiary structure of the proteins can be identified by using X-ray crystallography and other techniques. Okay, in the X-ray crystallography, even myself, I have not seen this technology, but what they do is they try to get the protein crystal, and they pass the X-ray beam, monochromatic X-ray beam through that crystal, and these crystals will diffract this X-ray beam, okay, depending upon the orientation of the electrons that are present around the various atoms. So the X-ray beam will be diffracted, which will be identified by a photographic film, and then ultimately it will be collected by a computer system. This nuclear magnetic resonance has having more advantage because it can study the three-dimensional structure of the protein in the solution, in the aqueous solution. Okay. So that's all regarding the sequencing of protein and regarding Prions disease. So, Anu, just tell me the name of the Prions disease in humans.
Ma'am, Creutzfeldt-Jakob disease.
In sheep, what is it known as? Sheep
Um,
name?
sheep is spike.
Scarpes disease, no?
Scarpes
Yeah.
disease.
And in cow?
Mad cow disease. Bovine
Yeah,
spongiform encephalitis.
encephalitis. So actually these are caused by the same infectious protein which is just passed on from one species to another species. No, just like COVID, it is passed from one species to another species. So in the same way, infectious protein particle passed on from one species to another species, resulting in the prions disease with different names. Okay, we'll continue the class on plasma protein. Okay. So can you see this slide? This one? This slide? Students? Anup, can you see this slide?
Yes, ma'am.
Okay. So let us come to the plasma protein. So what is plasma protein? You know blood, isn't it? So what is the composition of blood? What do you think? What are the things which are present in blood? So in the blood you have got the blood cells, isn't it? Like erythrocytes, RBC, WBC, and the blood platelets. No, erythrocytes, leukocytes, and thrombocytes. Other than that, you have got hormones, you have got enzymes, no, you have got plasma proteins, you have got minerals, vitamins, everything is present in the blood. Clear? Okay, so you know what is the difference between plasma and serum? Anyone? Anup, you know what is the difference between a plasma and a serum?
Ma'am, serum is plasma without clotting factors.
Okay, without clotting factors, and plasma is
With clotting
yeah,
factors.
with clotting factors. Okay, so when you add an anticoagulant into our blood, okay, and centrifuge the blood, you will get a clear straw-colored supernatant, which is known as plasma. But without adding an anticoagulant, if you allow the blood to clot for some time, and then you centrifuge the blood, then the supernatant is known as serum. So remember, serum and plasma difference is serum is without anticoagulant and plasma is with anticoagulant. Okay, so in the serum, all the clotting factors have been utilized, but in the plasma, the clotting factors are still there because you are adding an anticoagulant. Clear? So this is a slide which shows the serum protein electrophoresis. It has been taught in the practical class, protein electrophoresis. Yes or no?
No, ma'am.
No. So in protein electrophoresis, what we do is we take a glass slide. Okay, in the glass slide, we layer the glass slide with agar. Okay, and then we put this glass slide in a tank which contains a buffer, And this tank is connected at both ends to the opposite electrode. One is the anodal side, another is cathodal side. Okay, so buffer tank, which contains a buffer and the two electrodes. Then what you do is you suspend this glass slide with the agar medium into the buffer, okay, which is present in the tank, and connect it to an electrical field. Okay, and then you apply a spot where the plasma proteins are being added. For example, this is the spot here towards the cathodal side. We will always add the spot towards the cathodal side, and then pass an electrical field, and these plasma proteins, depending upon the molecular size and the charge they carry, they will migrate at different distance. Since albumin is the lowest molecular weight, okay, and it is having a highest charge, it will migrate faster when compared to the rest of the plasma protein towards the anodal end. So when you perform this technique is known as electrophoresis. Phoresis means movement and electro means electrical field. So it is the movement of the plasma protein in an electrical field is known as electrophoresis, and it is performed on an agar medium which is layered on the surface of the glass slide. Okay, so you add the plasma proteins, mixture of plasma proteins at one end of the glass slide, and they will migrate depending upon their molecular size and the charge they carry. So the first one to migrate towards the anodal end, remember it is albumin, then comes alpha-1 globulin, then alpha-2 globulin, beta globulin, and gamma globulin. Okay, so same thing when you see the peak, when you see the height of this various peaks, then you can see that the concentration of the albumin is highest. Okay, so concentration of the albumin is highest, so it will form a bigger band. You will get a bigger band when the concentration of the since the concentration of the albumin is highest in the plasma, that's why you will find a bigger band. Then the next band that you see is alpha-1 globulin, remember, then next is alpha-2 globulin, then beta globulin, and gamma globulin. So the concentration of alpha-1 globulin is lowest in the plasma, okay, when compared to alpha-2, beta, or gamma. The most abundant is albumin, then gamma globulin, okay, then alpha-2, beta, and alpha-1. Clear? So this is how the proteins will migrate in an electrical field, and they form different bands. The first band towards the anodal side, remember, it is albumin. Then you get alpha-1 globulin, then alpha-2 globulin, beta globulin, and gamma globulin. Okay, sometimes what happens, instead of adding blood or the plasma, you have added the CSF and you have performed electrophoresis, then you get a band which is ahead of this albumin, which is known as pre-albumin. In this slide, there is no pre-albumin band, but sometimes we can see a pre-albumin band, okay, which is more towards the anodal end. And this happens when you add CSF, or when the separation of the plasma protein is too good, then you can sometimes see the pre-albumin. Okay, the protein which is present in the pre-albumin is, remember, transthyretin. Transthyretin, which is required for the transport of thyroxine. Okay, transthyretin, sometimes it can form a band ahead of albumin, which is known as pre-albumin, and this transthyretin is required for the transport of the hormone thyroxine. Yeah. So since you can see the band of the albumin is thickest, that means the concentration of albumin is highest when compared to other plasma proteins. Okay, so the total protein concentration, all the analytes, remember, in biochemistry is usually in milligram. Protein is the only analyte which is expressed in grams per 100 ml or grams per deciliter or grams per cent. Okay, these are all same. So total protein is the only analyte, remember, which is expressed in grams. All other biochemical analytes are expressed either in milligram, microgram, nanogram. Clear? So the total protein concentration in the blood, remember, it is around 6 to 8 gram per cent, the normal value. Albumin, the normal value is around 3.5 to 5 gram per cent. Globulin concentration is less in the plasma. This one normal value is around, it is between 2.5 to 3.5 gram per cent. Clear? So this is usually asked in the exam. what is the normal value of total protein, what is the normal value of albumin, what is the normal value of globulin. So remember, in the plasma, the major proteins are albumin, globulin, and fibrinogen. Okay, you can write down the fibrinogen normal level is around 200 to 400 milligram %. Okay, so fibrinogen is 200 to 400 milligram %. Now albumin, the first band that you saw in towards the anodal side is albumin. So albumin, remember, all the plasma proteins in our body is synthesized in which organ? Whatever proteins are present in the body, where is it synthesized? In which organ? Chandani?
liver.
In liver. Okay, can you name one protein which is not synthesized in the liver? Anup, bolo. Any protein which is not synthesized in the liver? All the proteins.
Hemoglobin.
Hemoglobin is not a protein, no. It is a conjugated protein. It is a conjugated protein which is made up of globin as well as the heme group. But the globin is synthesized in the liver. One protein which is not synthesized in the liver, which is synthesized by the WBC. Any protein that is synthesized by the lymphocytes? Immunoglobulin, isn't it? It is immunoglobulin. Clear? So all the proteins, remember, they are synthesized in the liver except immunoglobulin, which is synthesized by the white blood cells, that is by the lymphocytes, mainly the B lymphocytes. Clear? The B lymphocyte synthesizes the immunoglobulin. So this albumin is synthesized, I will not go through every slide. So albumin is synthesized in the liver, and it is the most abundant protein present in the plasma. The molecular weight of albumin is 69,000. Hemoglobin, it is lesser than albumin, 65,000. That's why whenever there is any injury to the glomerular membrane, albumin as well as hemoglobin comes out in the urine. Okay, so what is the function of albumin? The most important function of albumin is maintenance of the osmotic pressure inside the blood vessel, which is known as oncotic pressure. Okay, the osmotic pressure exerted by the plasma protein is known as oncotic pressure. So how will you define osmosis? Osmosis is the movement of the solvent from its higher concentration towards the lower concentration, isn't it? So this albumin, remember, it maintains the oncotic pressure in the blood. Then albumin is also required for the transport of few substances in our body. For example, bilirubin. Okay, bilirubin is synthesized in the liver. Then it has to be, bilirubin is not in the liver, bilirubin is synthesized in various tissues, and then it has to be transported to the liver for conjugation. Okay, so bilirubin is insoluble, so it has to be conjugated with albumin. Okay, and then it is transported to the liver. So it helps in the transport of hydrophobic substances like bilirubin, then non-esterified fatty acids, drugs like phenytoin, dicumarol. Then albumin also has got a buffering action. And remember, the buffering action of any protein is because of the histidine residue, because the pI of histidine, isoelectric point of the histidine, is near to the physiological pH. Okay, so the buffering, albumin has got the buffering action, plus albumin has got a nutritional value. So let us see what is the role of albumin in maintaining the oncotic pressure. So suppose this is a capillary. Okay, this capillary, this is the arterial end of the capillary, and this is the venous end of the capillary. Towards the arterial end of the capillary, the blood pressure is around 35 millimeter of mercury, and the oncotic pressure exerted by the plasma protein is 25 millimeter of mercury. So the difference is 10 millimeter of mercury. That means your hydrostatic pressure is more than the oncotic pressure. So what will happen? The fluid molecules will come out from the arterial end of the capillary towards the, in between the tissue spaces. Yes or no? But towards the venous end, you see that the oncotic pressure will be same. No, but the hydrostatic pressure is lower, that is 15 millimeter of mercury. So what will happen? The water molecules, whatever was there in the in between the cells, it will move into the capillaries. Okay, so that is how the albumin maintain the oncotic pressure. But what happens if the albumin concentration is low? If the albumin concentration is low, the osmotic pressure or the oncotic pressure exerted by the albumin will be low. No? So it may be lower than the hydrostatic pressure. So whatever fluid is accumulated in the tissue cannot enter back into the capillaries, and then it will lead to a condition known as edema. Clear? Any confusion? If there is confusion, please ask. Vivek, confusion है या नहीं?
No,
Ma'am,
ma'am.
can you repeat once?
Yeah. तुम लोगों को participate करना पड़ेगा, okay? In the capillary end, what is the hydrostatic pressure? In the arterial end of the capillary, what is the hydrostatic pressure? बोलो विवेक, कितना है hydrostatic pressure? Arterial end में what is
35 mm.
Yeah, 35. And what is the osmotic pressure, the oncotic pressure exerted by the albumin?
25 mmHg.
25 mm. So here what the hydrostatic pressure is more than the oncotic pressure, isn't it? Yes or no?
Yes, ma'am.
So what will happen to the fluid that is present inside the blood vessel? What will happen to the fluid that is present in the blood vessel? Since the hydrostatic pressure is more than the oncotic pressure, it will come out into the tissue spaces. Yes or no? In the arterial
Yes,
end.
ma'am.
But towards the venous end, क्या होता है? Your hydrostatic pressure is lower than the oncotic pressure. No? Yes or no? Hydrostatic
Yes,
pressure is
ma'am.
lower than the oncotic pressure, so the solvent or the water molecules will move from the tissue spaces inside the capillaries, because osmosis, you know, it is the movement of the solvent particle from its higher concentration towards its lower concentration. So the water molecules will move from the tissue spaces into the capillaries. But if your albumin concentration becomes low because of various clinical conditions, the oncotic or the osmotic pressure will be low. Okay, so whatever fluid is accumulated in the tissue spaces, it cannot enter inside the blood capillaries. As a result, it will lead to a condition known as edema, which is nothing but the collection of fluid in the tissue spaces. Clear? Okay? Anup, समझ में आया?
Yes, ma'am.
Okay. ठीक है. We'll move ahead. So this is how albumin maintains the osmotic pressure. We can also say oncotic pressure. Okay, and what is the role of this one? What happens when the albumin concentration decreases? Okay, so it maintains the colloidal osmotic pressure. Second function is to transport the hydrophobic substances. For example, bilirubin, you have got non-esterified fatty acids, drugs. Then albumin has got a buffering action because of the histidine residues, and then it has got a nutritional value. Okay, albumin concentration is reduced in conditions like malnutrition. Okay, example of malnutrition is protein energy malnutrition. So in such condition, because there is decreased intake of the protein, that's why the albumin concentration also decreases. In nephrotic syndrome, there is loss of albumin, and then there will be an edema around the face in nephrotic syndrome. Remember, okay, the edema around the face occurs in nephrotic syndrome. In case of liver diseases or liver failure also, there is an edema, and the edema occurs it is inside the abdominal cavity, leading to a condition known as ascites, and then edema may occur in the dependent area. In congestive cardiac failure also, remember the edema is in the feet, the dependent area. Clear? In nephrotic syndrome, the edema is in the face. So albumin is reduced in conditions like malnutrition, then number second in nephrotic syndrome, okay, because of increased excretion. What is cirrhosis? Cirrhosis is liver failure. So if the liver fails, because liver is the organ where the albumin is synthesized, so a failure of the liver will lead to decreased albumin concentration. Then in case of congestive cardiac failure, there is edema and decreased albumin concentration. So this one I have already told what are the functions of albumin. The normal albumin level 3.5 to 5 gram%. Other things you can read by yourself. There is a condition known as microalbuminuria. Okay, what do you mean by microalbuminuria? When the albumin excretion in the urine is between 30 to 300 milligram. Okay, 30 to 300 milligram. When the excretion of the albumin in the urine is between 30 to 300 milligram, that condition is known as microalbuminuria. Clear? And this microalbuminuria occurs in case of diabetes mellitus and in case of hypertension. So it is an indicator that the person may develop renal failure in future renal failure. So what is microalbuminuria? When the albumin concentration in the urine is between 30 to 300 milligram per day. Okay, so albumin, when you perform an electrophoresis, the albumin is the first band and it is the thickest. Okay, the percentage is 55 to 65. Then alpha 1, 2 to 4%, alpha 2 is 6 to 12, beta globulin 8 to 12%, almost same thickness. Then gamma globulin 12 to 22%. Now let us see what are the proteins which are present in the alpha 1 region of the electrophoresis. This is very important. Sometimes it is difficult to remember also. So whatever you know, whatever protein, name of the protein you know, which has got the prefix of alpha 1, in that way you can remember what are the proteins which forms the alpha 1 band. It is alpha 1 acid glycoprotein, clear? Then alpha 1 lipoprotein. What is the other name of alpha 1 lipoprotein? Anyone? You have already learned about lipoproteins, isn't it? You have got HDL, VLDL, LDL, no? IDL. So what type of lipoprotein is alpha 1 lipoprotein? Anyone?
High density lipoprotein.
Yes, good. So high density lipoprotein is alpha 1 lipoprotein. So high density lipoprotein, remember in electrophoresis, it will occupy the alpha 1 band. So what are the proteins which forms the alpha 1 band? Remember alpha 1 acid glycoprotein, alpha 1 lipoprotein, alpha 1 phytoprotein, alpha 1 antitrypsin, and alpha 1 globulin inhibitors. Other than that, you have got thyroxine binding globulin. Okay, so it is difficult to remember, but at least you remember at least 2 or 3 names. Okay, you can remember alpha 1 lipoprotein, alpha 1 antitrypsin, very important. Deficiency of alpha 1 antitrypsin can cause emphysema. Okay, then alpha 1 phytoprotein, it is a biomarker for liver cancer. Okay, it is a biomarker for liver cancer in the this one. In case of fetus, it is a biomarker for neural tube defect. Okay, neural tube defect. And in case of adults, it is a biomarker for hepatic cancer. In the alpha 2 region, you have got the proteins like alpha 2 glycoprotein. The spelling is wrong. Ceruloplasmin. Okay, what is ceruloplasmin? It is a protein which stores copper. So when there is deficiency of ceruloplasmin, the copper gets deposited in various organs, like in the lens, it is deposited in the brain, and it leads to a condition known as Wilson's disease or Wilson's hepatolenticular degeneration. So all these proteins like alpha 2 glycoprotein, ceruloplasmin, haptoglobulin, plasminogen, prothrombin, and alpha 2 microglobulin, all these proteins that occupy the position of alpha 2 band. Haptoglobulin, remember, it is a protein which complexes the hemoglobin, and it does not allow the hemoglobin to be released into the urine. So haptoglobulin, it preserves the hemoglobin molecule whenever there is lysis of RBC. Clear? So what is ceruloplasmin here? You cannot remember everything, but at least you can remember those protein which starts with the prefix alpha 2 macroglobulin, and alpha 2 glycoprotein. Remember, this alpha 2 macroglobulin is increased in case of nephrotic syndrome. Okay, it is increased in case of nephrotic syndrome. Ceruloplasmin, what is it? Ceruloplasmin, you will be learning later again. It is a copper storage protein. Okay, it stores the copper. Whenever there is deficiency of ceruloplasmin, the copper will get deposited in various organs, leading to a condition known as Wilson's disease. What is the role of haptoglobulin? It complexes with hemoglobin, and it does not allow the hemoglobin to be excreted into the urine in case there is breakdown of RBC. Clear? This prothrombin, plasminogen, these are all required for coagulation. Then what are the proteins which are present in the beta globulin? You have got the beta lipoprotein, which is nothing but LDL. Okay, then remember this C-reactive protein. C-reactive protein, it is also present in the beta region. I will tell in detail regarding the C-reactive protein. What is transferrin? It is a protein which helps in the transport of iron. Okay. Hemopexin, it complexes with RBC. Then you have got the complement proteins and beta microglobulin. Okay, so the proteins which are present in the beta band, at least you can remember beta lipoprotein and beta microglobulin. Then in the gamma band, what is present? Mainly immunoglobulin. Okay, remember in the gamma band you have got immunoglobulin. So again we will repeat the thing. So this is an albumin band. So it is only the albumin protein. So this albumin band is decreased, remember, in nephrotic syndrome. Okay, so the peak of this albumin band will be decreased in nephrotic syndrome. The next band is the alpha-1 band. What are the proteins which are present in alpha-1 band? Alpha-1 lipoprotein, you can remember. Then alpha-1 antitrypsin, alpha-1 phytoprotein, isn't it? Then alpha-2. In the alpha-2 region, what are the proteins present? In the alpha-2 region?
Alpha 2
Alpha 2
glycoprotein,
glycoprotein,
Okay, any other?
alpha 2 macroglobulin,
Macroglobulin, okay. And
ceruloplasmin,
ceruloplasmin. In the beta band, what is present? Beta band. C-reactive protein is present in which band?
beta globulin.
Yeah, beta band. No, in the beta band, C-reactive protein is present. Okay, so C-reactive protein is an acute phase protein. So how will you define acute phase protein? The proteins which are increased or decreased in acute conditions like injury, inflammation, no, or after surgery or during neoplasia, these proteins are known as acute phase protein. Okay, so any proteins that are increased or decreased in acute conditions like inflammation, injury, etc. are known as acute phase protein. Their concentration may be increased. In that case, we call them as positive acute phase protein. When the concentration is decreased, they are known as negative acute phase protein. So remember, C-reactive protein is a positive acute phase protein. What is present in the gamma region? This gamma,
Immunoglobulins.
yeah, immunoglobulin. So remember this albumin band, it is decreased in nephrotic syndrome. The alpha-1 and alpha-2 bands, they are increased in acute inflammation, okay, or acute infection. Alpha-1, alpha-2, beta, okay, and the gamma band all are increased in the chronic infection or chronic inflammation. Then there is decrease of this gamma band in primary immunodeficiency. Okay, and sometimes in the gamma band, you may see a sharp band here. Okay, like this. This whole thing is gamma. So you can see a sharp band in the gamma region. That band is known as M band or multiple myeloma band. Okay, so this multiple myeloma band, remember, it usually occupies the gamma region, but sometimes it may be present in other regions only, but regions also. But maximum time, the M band is present in the gamma region, and M stands for multiple myeloma band. It is present in the plasma cell tumor, which is nothing but multiple myeloma. Okay, so multiple myeloma M band is present in the gamma region, and gamma band is decreased in primary immunodeficiency. Alpha-1, alpha-2 bands are increased in acute inflammation. Alpha-1, alpha-2 and beta band, all three are increased in chronic inflammation. Albumin band is decreased in nephrotic syndrome. So these are some of the clinical conditions where these bands, the concentration or the density of this band may be increased or decreased. Clear? This alpha-1 antitrypsin, remember, it is present in the alpha-1 region, and deficiency of this alpha-1 antitrypsin leads to a condition known as emphysema. Please note it down. Alpha-1 antitrypsin deficiency leads to a condition known as emphysema. Only some important points I will tell you because the chapter is very long. So I have already told what are the proteins which are present in different regions. So fibrinogen, okay. Fibrinogen is also a plasma protein. The concentration is in milligram. On the other hand, albumin and globulin concentration is in gram. Some transport proteins, let us see the examples of transport protein. Albumin is a transport protein. It helps in the transport of bilirubin, calcium and various drugs. Transthyretin, which forms a pre-albumin band, it helps in the transport of thyroxine. Then thyroxine binding globulin also helps in the transport of thyroxine. Retinol binding protein helps in the transport of vitamin A. Okay, transcortin helps in the transport of cortisol. Haptoglobulin for heme. Hemopexin is for heme. Transferrin helps in the transport of iron. Okay, and this LDL, HDL helps in the transport of cholesterol or cholesterol esters. So I've already told acute phase proteins are the proteins which are increased or decreased in inflammatory or neoplastic conditions. And example is C-reactive protein, ceruloplasmin, haptoglobulin, alpha-1 antitrypsin or fibrinogen. These are positive acute phase protein. Okay, so if they ask you name one positive acute phase protein, at least few examples you can remember. C-reactive protein is a positive acute phase protein. Its concentration increases during bacterial infection. Okay, and another is alpha-1 antitrypsin, which increases during emphysema. Ceruloplasmin increased, okay, during any acute infection or inflammation. So this C, sometimes they ask you, the C-reactive protein, what this C stands for. Okay, the C stands for the polysaccharide which is present in the capsule of the pneumococci. Okay, so this protein, it reacts with the polysaccharide which is present in the capsule of the pneumococci. Polysaccharide or carbohydrate, you can say. C stands for carbohydrate. So this protein reacts with the carbohydrate which is present in the capsule of the pneumococci. That's why it is known as C-reactive protein. Okay, and the C-reactive protein is increased in bacterial infection, and sometimes it is used to distinguish between bacterial and viral infections. Then sometimes in our lab we have got high sensitive CRP, and this high sensitive CRP is increased in case of myocardial infarction. So in myocardial infarction there are so many proteins which are increased, isn't it? One is CK, then myoglobin, then you have got troponins, and then nowadays we have got high sensitive CRP which is increased in myocardial infarction. Then alpha-1 antitrypsin, remember, you cannot remember everything. It is increased in case of emphysema. So alpha-2 macroglobulin, some important points, increased in nephrotic syndrome. Okay, so in nephrotic syndrome, if you get a sample of nephrotic syndrome and you perform electrophoresis, the albumin band will be decreased and the alpha-2 band will be increased in nephrotic syndrome. Clear? Then negative acute phase protein, the concentration of this protein decreases during acute infection or inflammation. Example is transthyretin, which helps in the transport of thyroxine. Okay, ceruloplasmin is the positive acute phase protein. It helps in the storage of the copper. How many coppers are it can bind per molecule? Remember, 8 to 6. And this ceruloplasmin is having an antioxidant property. It has got the enzyme activity known as ferroxidase. Okay, they used to ask in the exam, ceruloplasmin is having ferroxidase activity. It converts the ferric to ferrous to ferric ion, and then it helps in the insertion of this ferric ion into a transferrin molecule. Transferrin is the protein which helps in the transport of the iron. So this ceruloplasmin is having a ferroxidase activity which converts the ferrous to ferric ion, and when the iron is in the ferric state, only then it can bind to transferrin. Transferrin is the protein which helps in the transport of iron. Clear? So deficiency of this ceruloplasmin can lead to Wilson's disease or hepatolenticular degeneration. Okay, so the symptoms will be related to the brain, to the liver. So haptoglobulin, I have already told, it binds with the hemoglobin and thus prevents the loss of free hemoglobin through kidney. Then we have got various clotting factors. I think this one will be taught in your physiology also. I will not go into these clotting factors. So deficiency of these clotting factors will lead to a condition known as hemophilia A or B. So that's all. Okay, regarding plasma proteins. So in the plasma proteins, you just have to know what are the major plasma protein: albumin, globulin, and fibrinogen. And when you perform electrophoresis of plasma protein, you get different bands: albumin, alpha-1, alpha-2, beta, and gamma. Sometimes before albumin, you get a band that is pre-albumin that occurs when the sample is CSF and not plasma. Okay, and there are so many proteins which can form this band, and importance of certain plasma protein like ceruloplasmin, what are the acute phase protein, some of the things you can study. Okay, that's all regarding plasma protein. In the next class, I will start protein metabolism. So that's all for today's class, students. Thank you so much.
Thank you, ma'am.
Okay, you please go through whatever has been taught in the class, okay?
Thank you, ma'am.
Okay, welcome.